So where do you have action potential and where do you have greater potential? That's what we're going to talk about now. So this is just showing you how in the action potential it keeps going, different from the greater potential. There are videos on A&P Flicks in mastering A&P that you should totally watch because it just tells you what I just told you. Okay, so it's a way to review it. So speed. Let's go back and talk about speed. Action potential can be quick or slow. Greater potential always slow. Action potential is going to be slow when there is no myelination on these neurons. It's going to be quicker when the stimulus is jumping, leaping from channel to channel. You see, and every time it leaps, you have a higher peak of depolarization, which keeps it going quicker. And this happens because in these areas here, what do you have? Myelin. Okay? They're wrapping from the, if it's in the central nervous system, from the oligodendrocytes. If it's in the peripheral nervous system, from the Schwann cells. Okay? They wrap the axon. Sorry. It's similar, but this is what happens when you have a voltage-gated channel, not necessarily myelinated. It could be myelinated or not. This is the representation of the myelin sheath. So when you have the myelin sheath, those voltage-gated channels, so voltage is going to open all these sodium channels, and it's going to keep depolarizing the whole membrane. And leaping, because you have a myelin sheath, is going to make this propagation quicker. In the greater potential, it's going to be more like this. Why? What's the difference between the action potential and the greater potential? In the greater potential, you don't have voltage-gated channels. You only have that initial chemically gated channel, the neurotransmitter binds to it, and you have the one propagation that happens slow and it's short length. Where do you have greater potential? In the dendrites. The receptors from the neurons don't need a huge propagation wave. So it's just that short, potential that allows the signal to be received from that presynaptic neuron to the postsynaptic neuron, and then it goes, that signal goes through the neuron. So that initial receptive part of the neuron, which is here, you see, doesn't allow a very long-lasting effect. So it's a really short event, okay? No voltage-gated channels, only chemically gated channels, okay? So, with that said, knowing that action potentials are long-lasting, only neurons and muscles can have them, that they usually happen in the axon, right? So this is showing you the axon where the action potential happened. And that gated potential usually happen in the dendrites and in the soma. We are ready to talk about the synapses. So what is the synapses? It's what happens before this propagation of the action potential in that one neuron that we're talking about. So the synapses is the stimulus that comes to this neuron that we're talking about from a presynaptic neuron. So a synapses is an event that happens between two different neurons, the presynaptic and the postsynaptic neuron. And they have some type of interaction that is not really physical because there's always a synaptic cleft. There is a space between the axon terminal, see the axon terminal here, from one neuron and whatever receptor part of the second neuron. Okay, so axon terminal from presynaptic neurons can bind to soma, to dendrites, or to axon hillox. Okay. They bind to different places. And that's where the stimulus happens. What is the stimulus, as we said? It could be a chemical stimulus, it could be an electrical stimulus. Chemical is when you have neurotransmitter being released, and we're going to talk about which neurotransmitters we could have. And electrical is when what is passing from one neuron to the other is just charge, not a chemical, okay? This is a video that you can see later that talks about how the synapses happen. There is a diagram here that may help me explain this to you. So a chemical synapse happens when you have, let me see if I can make this bigger. When you have calcium going in, where is this? What's that? Axon terminal, right? From the presynaptic neuron. So this is an axon terminal that is quote unquote touching the postsynaptic neuron. You see how it's not touching? Because you have this space here that is called synaptic cleft, okay? So when calcium goes in, these vesicles with neurotransmitter are fused to the plasma membrane, and the cargo or the neurotransmitter are released by exocytosis. These transmitters bind in channels that open ion flux, right? And then you generate a graded potential or an action potential. Okay, usually in this case it's a greater potential because it's where you're receiving it. And then the action potential is going to happen after this greater potential happens. In an electrical synapses, these are less common, but usually you find in some brain regions that are responsible for eye movement or in the hippocampus, areas that are involved in emotions and memory. It's also very abundant in embryonic nervous tissue. Okay, so these are just electric charges that are passed from one neuron to the other. With that, there is a very nice table in the end of the book that is just going to summarize and give some more details about the difference between graded and action potential. But mostly what I need you to know is speed, where it happens, and type of receptors associated with that, okay? Neurotransmitters. You can classify, so these are those molecules that are released in the synaptic cleft. They can be classified by their chemical structure or by their function. Acetylcholine is a very common, and frequent one. This is how you write the acronym for acetylcholine, ACH. So you're going to see that, like we did in the muscles. Released at neuromuscular junction and released in other situations that we're going to see. Used for autonomic nervous system and some central nervous system neurons. Then you have other classification, other structures that are classified by structure. Biogenic amines, catecholamines, and indolamines. So you have dopamine, norepinephrine, epinephrine. These are catecholamines made from tyrosine. And indolamines, serotonin, histamine. These are other neurotransmitters that can play roles in emotional behavior and biological clock. Amino acids can work as neurotransmitters. Glutamate, aspartate, glycine, GABA. Okay, so these are all amino acid, very tiny molecules that have huge effects when they're released in the synaptic cleft. Neuropeptides like substance P related to pain, endorphins related to pain reception or perception, and some gut-brain peptides, something that regulates neuropeptides that regulate digestion on the enteric nervous system. You also have ATP that can be considered a neurotransmitter. And adenosine is a very potent inhibitor in the brain. So it's another type of transmission that is inhibitory. Okay? There are other substances like gases and lipids that can also work as neurotransmitters. And the effects of these drugs, like I said, can be excitatory or inhibitory. I am not going to ask you which one is what. I just wanted to mention this because later we are going to talk about situations where you're inhibiting an action potential, situations where you're exciting one. Okay? That's it. we're done. So please review resting potential, action potential, and graded potential so you have that very clear in your mind. And I'll talk to you when we talk about next chapter, which is the structures of the central nervous system. Bye.